Hi guys, in this video we're going to talk about oxygen transport. Oxygen transport in mammals occurs primarily by two main proteins, myoglobin and hemoglobin. Now what I've drawn here is a very schematic view of myoglobin. We'll look more detailed at the structure in lecture. Myoglobin is a folded protein, has lots of different alpha helices. Let me just write out myoglobin here. It consists of approximately 150 amino acids. And in addition to the amino acid chain, there's also a cofactor that's bound. I'm just going to write this as a line, and I'll put in here Fe plus. It's actually 2 plus. So there's an iron bound to this cofactor. If I were to sort of draw this out now in a little bit more organic chemistry way, I won't draw the entire structure of the heme group, but this cofactor is called heme. And it has 4 nitrogenous containing ring structures. They're actually based off the structure of the histidine side chain, which is called imidazole. But right in the middle of all of those nitrogens, there's going to be an iron, okay, an iron atom. And so that cofactor, and there's a lot of other stuff kind of surrounding this, holding it together. But now envision that cofactor embedded in the middle of this myoglobin protein chain. So myoglobin, myo means muscle. This is the primary oxygen transport protein that's found in muscle. Okay, and it has a very high affinity for oxygen. Okay, so when oxygen binds to myoglobin, it's actually going to be binding to the iron. So if we put O2 here, it's actually going to be binding right there onto the iron atom. So this helps explain why certain anemias, particularly those related to iron deficiency, occur. If you don't have enough iron to populate the heme groups of your myoglobin, you're not going to have efficient oxygen transport. So myoglobin is a single polypeptide chain, again, about 120 amino acids. And the key thing to remember is it has a very high affinity for oxygen. So let's talk about the second main protein involved in oxygen transport in mammals, and that is the molecule called hemoglobin. Now hemoglobin actually is a tetramer of 4 different polypeptide chains, so it actually exhibits quaternary structure. It contains 2 of what are called the alpha chains, and these are 141 amino acids, whereas the beta chain is a little bit longer, 146 amino acids. Now I won't try and draw what this entire structure looks like, but I'll just show it sort of schematically here. These are 4 separate polypeptide chains, 2 alphas and 2 betas. Each one has a heme in it. Okay, and each of those hemes has an iron. Okay, so there's actually 4 different binding sites for oxygen to bind on hemoglobin. Now hemoglobin, because it has this tetrameric structure and exhibits quaternary structure, does not have quite as high affinity for oxygen, and it exhibits what's called cooperative binding. So let's talk about looking at the binding affinities of both myoglobin and hemoglobin, and to do that, I'm going to introduce you to a type of graph called a percent saturation or affinity curve. Now on the axes, let's go ahead and just label those. I'm going to have what's called the partial pressure of O2, and that's listed in millimeters of mercury. You can think of this essentially as the amount of oxygen dissolved into the blood. All right, and I'll just list out some numbers here. We'll say 20, 40, 60, 80, and 100 millimeters of mercury. Okay, so this is again the partial pressure of oxygen. Now on the y-axis we have percent O2 saturation of a particular molecule. Now we've talked about myoglobin in the previous section, and now I'm talking about hemoglobin. Let's look at myoglobin's saturation curve. So at even very low pressures or low amounts of dissolved oxygen, the percent saturation, and let's just put say 100% up here and 50 here, if I were to draw the curve for myoglobin, it actually is going to look kind of like this. Okay, this is what's called a hyperbolic binding curve. Okay, and it essentially goes out to infinity here. Can't get more than 100%. But even at very low partial pressures of oxygen, okay, even down at say 20 millimeters of mercury, I have close to 50% saturation. When I get up to 40%, I'm up at say 90 to 95%. So that's called a hyperbolic binding curve, and that would be myoglobin. Now in contrast to that, hemoglobin has a very different looking curve. And the reason for that is because of what's called cooperative binding and the fact that you've got these 4 different subunits sort of communicating with each other. So initially, hemoglobin has very little affinity for oxygen. So even at fairly low pressures, you have fairly low affinity. But as we start to get higher and higher pressures, we then get a curve that's now going to look more like this. This is called a sigmoidal binding curve, and this would represent hemoglobin. Okay, so at very low pressures, down around 20 or 40 millimeters of mercury, you can see that our percent saturation for hemoglobin is maybe only 20%. When you start getting up to about 60, you start approaching 50% saturation, and then you have to go to even higher pressures to get your percent saturation for hemoglobin to approach 100%. All right, so to wrap up our introduction then for oxygen transport, I'm going to just draw a very, very crude schematic diagram of mammalian circulation. So here we have our lungs, and we have blood vessels that are infiltrating the lungs. When we inhale, we're going to be bringing in oxygen. Okay, so oxygen is going to be coming in. Around the alveoli of the lungs, we have basically the small capillaries that are going to be containing our red blood cells that contain hemoglobin. So in our blood and red blood cells, we have large amounts of hemoglobin. Now, as we inhale, the partial pressure of oxygen is going to increase quite a bit. So our hemoglobin will pick up O2. So O2 is going to now bind because the pressure is very high. Now it's going to then flow through the arteries and be delivered then ultimately to the muscles. Now I said that muscle tissue contains a lot of myoglobin. Myoglobin you can think of essentially as an extremely efficient oxygen sponge. So as the capillaries containing our red blood cells flow by with hemoglobin, they're going to take that oxygen that's bound to hemoglobin and they're essentially going to suck it off of the hemoglobin. So myoglobin is going to sort of grab a hold of it. And again, the reason for that is because it has high affinity for oxygen at all partial pressures. So in the lungs, we have a very high affinity for oxygen with hemoglobin because the pressure is very high. But as we start to get out to the muscles, the concentration of oxygen or the partial pressure becomes quite a bit lower. And we know from our previous diagram that at low partial pressures, hemoglobin has a very low saturation percent. So myoglobin, again, is going to basically suck the oxygen off of the hemoglobin. Hemoglobin, okay, will then recycle back to the lungs through the veins and then get reoxygenated. So the bottom line is that the muscle tissue is where we really need the oxygen so that we can go through oxidative respiration. And the hemoglobin is essentially just acting as a transporter to get the oxygen from our lungs ultimately delivered to our surrounding tissues. All right, so again, that's a very crude diagram. We'll cover this more in lecture, but I hope that helps on oxygen transport.